Hello everyone, this is our final video for genetics for this unit, and it's not going to be very long, so here we go. I'm actually going to direct you to a bunch of other videos online, or actually possibly to do some research in a second. So this is going to be a very short video indeed. So this is the idea of genetic testing. Genetic testing is a growing field of study and action in biology. Certainly genetic testing is now used throughout, even with our current coronavirus pandemic. There's lots of genetics being studied to see connections between the disease phenotypes and genes. There'll also be there also a lot of interest in studying the genetics of the virus itself. So genetic testing is a huge market, huge aspect of modern biology. And this little 2 pages are not nearly sufficient enough to get into it. So that's where we'll be doing some independent research. So the first place that we do genetic testing is through karyotype analysis. This is an older technique that's been done for many, many years. And that's used typically to identify non-disjunctions like in Down syndrome or triple X, Turner syndrome, and others. And you can see some introduction into how the karyotype is made. Here we actually went through this in the previous video, but now we have a picture to go with it. So let's talk about that. So first, you get a sample of the individual's blood cells, body cells, and you need white blood cells because those ones have nuclei. The red blood cells do not. And then, if you want to study a fetus, as many will when they are concerned about something and they need a genetic test, then you would be looking for some amniotic fluid, and that's through an amniocentesis, which we'll talk about in a second. So these cells are then divided and are allowed to divide and grow in a solution in the lab. So you can see here the removal of the red blood cells, and then these cells there's a chemical called Clochinsin. Clochinsin? Anyways, someone would correct me if we were in class, but too bad. So this stops the cells in their metaphase. And then we add some water, and when you add water to cells, they absorb the water through osmosis, and the cells get very large, and then the white blood cells will settle out, and then they can be spread upon a slide and fixed with some alcohol and stained. And now that they're stained, some of those metaphase cells will have burst. Those cells that burst don't have a nucleus, and when they are when they are stained onto this plate, they'll be actually spread out. And if you scan the whole slide, at least one of the cells is likely to be sort of organized in a nice enough way that you can take a photograph and then literally take a pair of scissors, probably done digitally now, and cut out each of the chromosomes and organize them, paste them out in size from largest to smallest, and analyze them. And if you have more or less chromosomes than you expect, then that would tell you that there is some sort of chromosomal disorder or change that's happened. And that's how a karyotype is made. Now the process of amniocentesis, and this I seem to get different numbers on this every year, but essentially what we have is an ultrasound up here. This uses sound waves that are bouncing off of the object on the inside and getting an image that way. It's the same idea as sonar. We'll guide a needle. This one has been blown up quite a bit to make it obvious, but that needle will be guided into the birth chamber there and extract some amniotic fluid. Now, ideally, the needle is so small that it will, when it pierces into the amniotic fluid, it will not disturb the fetus at all. And it won't even disturb the sac. It's going to be such a small, thin needle that it should not damage the sac at all. Now, there is the case where 1 in 200 pregnancies can be terminated by accident from this amniotic amniocentesis. And this could be from a variety of reasons. Possibly the sac is thin, or there's a jostling of the needle, or any sort of disturbance of the amniotic sac can cause a miscarriage to occur. So that's why this does come with some sort of irregularity from externally. They might recommend that an amniocentesis be done, especially if the family has a history of genetic disorder, or there's other signs that the fetus is having issues. Something like Down syndrome, the fetus might be undersized. They can detect irregularities in the heartbeat, and then they'll have other reasons for attempting to do the amniocentesis. Some fetuses are more mobile, especially early in the pregnancy. The fetus can move around quite a bit, and so there's a risk that the needle is going to be close to that baby who's moving. Now, the idea of an ultrasound is simply that it's sound waves that bounce off of the internal organs of the pregnant woman, and they don't disturb the baby at all. They are used without effects on the child at all. And that's one of the first places they're going to look for abnormalities. They'll look for changes in how the fetus is developing, and then they'll use that as a reference point for whether they should investigate further. Umbilical cord sampling is also an option where they can sample the fetus's blood. This blood is taken directly from the umbilical cord, and it can be used to test for other disorders like hemophilia and anemia, but it has a higher miscarriage rate, so it's much harder to do. Instead of just getting the fluid, you're actually looking to get in on that umbilical cord, and of course that's a much harder target to hit, and so that's very dangerous. So I'm going to leave that there. It's a sort of a strange place to end our genetics unit, I think, but that's the end of the unit. So we've covered a lot of different ideas. Please go back and review any videos you need to review. Otherwise, I will be moving to producing a final assessment that will be posted shortly, and also possibly some investigations into different aspects of genetic testing and what we are doing nowadays in the modern genetic testing lab. All right, with that, I leave you to work on your stuff. Okay, have a great day.